Let's go on, and our next speaker is Marta Maleszewska from Poland, and the title of her presentation is DNA Methylation and Gene Expression Analysis reveal the MRTA2 as a new important factor in high-grade glioblastoma and glioblastoma-derived spheres. So, Marta, please go on.
Can you hear me?
Yes, we can hear you.
You can hear me, and you can see my
screen. You can
screen,
see your presentation.
Okay, good. So thank you very much for this. I would like to thank the organizers very much for this invitation and this opportunity to present this work that concerns the MRTA2 as a new important factor in glioblastoma and glioblastoma-derived spheres. And it was discovered by DNA methylation and gene expression analysis. I'm research assistant at the Laboratory of Molecular Neurobiology at the Lensky Institute in Warsaw, in the lab that is headed by Professor Bożena Kamińska. So, glioblastomas, I think I will skip this because Victoria presented all the problems perfectly. So, her conclusion that we need new targets and new therapeutic strategies is very important to us. According to WHO classification, glioblastomas are divided into 4 malignancy grades. And the most, I will just try to, okay, this does not work here. Okay, anyway. So with pilocytic astrocytomas being benign tumors that are diagnosed mostly in children, and primary and secondary glioblastomas being the worst and with the poorest prognosis, 4th grade tumors. This WHO classification was mostly based on the histopathological diagnosis of patients. However, in the last few years, molecular diagnosis also joined to the diagnostic and grading system. And the most prevalent mutations that were selected for this stratification concern histone variant mutants for pediatric glioblastomas and isocitrate dehydrogenase mutations in adult tumors of lower grade. Wohowever, for the grade 4 glioblastomas, we don't really have any molecular markers and molecular targets that would be efficient so far. So, interestingly, most of these molecular targets, molecular diagnostic tools that are included in the classification are somehow epigenetic related, and epigenetics mechanisms are responsible for changes in gene expression that can be inherited through cell division, but are not associated with changes in DNA sequences. And one of the mechanisms of the epigenetic mechanism that is best described is DNA methylation. And DNA methylation concerns, in mammalian cells, DNA methylation concerns cytosines that are present within CpG dinucleotides. And in general, DNA methylation is associated with transcriptional inhibition. In cancer, DNA methylation, the aberrations in DNA methylations are quite well described, and we can consider them in a local changes, so concerning specific genes, and we observe both. So aberrant loss of DNA methylation that can lead to activation of oncogenes, or aberrant methylation of tumor promoters that can in fact inhibit expression of tumor suppressor genes, for example, but also general and global changes in DNA methylation in cancer cells well described. And what comes out from those studies is that in general we can see in cancer cells, we can see aberrant methylation of some crucial gene promoters that lead to gene expression inhibition. But on the general scale, we can see a global hypomethylation of the genome in cancer cells. So the aim of our studies was to identify genes that are aberrantly expressed in high-grade glioblastomas due to changes in DNA methylation. And for this purpose, we isolated DNA and RNA from glioblastomas, so the high-grade glioblastomas grade 4, and pilocytic astrocytomas, so those benign tumors of grade 1, subjected it to hybridization to DNA methylation and gene expression microarrays. We identified differentially expressed and differentially methylated genes in those groups, in those gliomas, and then intersected the results to find the genes that are expressed, that are aberrantly expressed in glioblastomas due to DNA methylation. So the analysis of the genome of those low and high-grade gliomas. First, we identified a number of genes that are hypomethylated and hypermethylated both in benign and high-grade tumors. And they are not, there is a bunch of genes that are intersected, but there are also some characteristic aberrantly methylated regions that are characteristic for low and high-grade tumors. Then we decided to analyze the distribution of the hypo and hypermethylated CpG sites in specific regions of the genome. So for this purpose, we divided the genome into CpG islands. So these are the regions that are enriched for CpG dinucleotides. Then we have CpG islands, shore, and shell. So the regions that are neighbor to CpG island, and the regions that are distal from CpG islands that we call open C. And as you can see, we could see aberrant distribution of hypo and hypermethylated sites in both pilocytic astrocytomas and glioblastomas. Interestingly, we could see an increase in hypomethylation. We could see an increased hypomethylation of CpG islands in low-grade tumors and hypermethylation in the open C. In contrast, in glioblastomas, we could mostly see hypermethylation of CpG islands and CpG island shores, so those closest to CpG island regions, and hypomethylation in open C and in shore, so in the distal regions from CpG islands. Further, we compared the DNA methylation and gene expression between those two grades of tumors, and we could see a number of genes that are differentially methylated in low grades and high grades tumors, and only a small number of genes that were actually differentially expressed between high grades and low grades. Further analysis showed that there were only actually 29 differentially methylated regions in those tumors that were hypermethylated in GBMs and hypomethylated in pilocytic astrocytomas. When we compared, analyzed the gene expression within those 29 differentially methylated regions, we discovered only 3 genes. Actually, we ended up with 3 genes that were differentially expressed due to DNA methylation in high-grade gliomas compared to low-grade gliomas, and those genes are DLX1 and DMRTA2. So these genes are methylated at the 3' UTR, and interestingly, this methylation of the 3' UTR correlates with higher expression of those genes in high-grade tumors. And the third gene was IRX2. We found the methylation in the gene body, and this correlated classically with IRX2 gene silencing in high-grade gliomas. So from those genes, the most interesting one seems to be DMRTA2, and that's because its expression actually correlates with worse survival of GBM patients. Actually, this gene from the literature data, we know that it is involved in early brain development as well as neuron glia sulfate switch. However, there are no data concerning the role of DMRTA2 in cancer. Therefore, we decided to deep in, and we first, thanks to the repository of glioma samples that we already collected in the lab, we confirmed that DMRTA2 is really is really highly expressed in GBMs compared to lower grade tumors and normal brain as well. And we also confirmed by MSPCR that there is a methylation of the DMRTA2 3' UTR region in high-grade gliomas, and there is no, and it's very rare in pilocytic astrocytomas. Next, we performed immunostaining for DMRTA2 protein in glioblastomas of different grades. And as you can see, we can see much higher staining, much stronger staining in glioblastomas compared to pilocytic astrocytomas. And what also was very interesting to us, that we could see an increased staining around of the DMRTA2 positive cells around blood vessels, as well as increased staining of small, as it was described by the pathologist, small cells with very high proliferative potential also display higher DMRTA2 positivity. Next, we decided to identify the DMRTA2 positive cells. What kind of cells express what are the cells that express DMRTA2? So first, we decided to perform double staining for DMRTA2 and OLIG2, which is a marker of oligodendrocyte precursor cells, and also for CD45, which is a marker of leukocytes, so immune cells. But we could not see any overlap between the stainings. So this means that DMRTA2 positive cells are not oligodendrocyte precursor cells and are not immune cells in the brain. Next, since we could see this increased DMRTA2 staining around blood vessels, we decided to perform a double staining for DMRTA2 and von Willebrand. And von Willebrand factor is a marker of endothelium. And as you can see here, we could not really see an overlap between von Willebrand and DMRTA2 staining. So the DMRTA2 cells are not really endothelium. However, they lie just next to the endothelium cells. And this led us to another hypothesis. And we performed another staining for DMRTA2, this time with von Willebrand and alpha-SMA. And alpha-SMA is a pericyte marker. Pericytes are mural cells for vascularization, and they support vessel formation in the brain. So as you can see, this time we could see a double staining of DMRTA2 and alpha-SMA, and von Willebrand cells are inside, that are surrounded by this double stained DMRTA2 positive and alpha-SMA positive cells. So this led us to conclude that DMRTA2 positive cells are alpha-SMA positive cells that could remain pericytes in the glioblastoma tissue. We also checked for DMRTA2 expression in glioma cell lines, and we could observe high expression at the mRNA, as well as at the protein level. In cell lines, both established cell lines and patient-derived cell lines. So the higher expression was present in the cell lines that in our hands are capable of formation. So this was a very interesting observation, that this DMRTA2 expression is higher in cell lines that form spheres. So then we decided to check the expression of DMRTA2 in spheres. And as you can see, we could observe higher expression of DMRTA2 in spheres compared to bulk cells for established and prime patient-derived cell line. And we also could see the increased expression at the protein level, which we confirmed by Western blot and by immunostainings. So then we decided to silence DMRTA2 and see if cells will be still capable of sphere formation. And as you can see here, we managed to silence the gene efficiently, and this actually led to inhibition of sphere formation capacity of LN18 glioma cells. And finally, I wanted to share with you some very preliminary data that we obtained recently in our laboratory. So as you remember, we could observe the DMRTA2 positive staining around blood vessels. And we could see that DMRTA2 positive cells are also alpha-SMA positive cells, so positive for pericyte markers. And pericytes are cells that support and sustain blood cell vessel formations. So we decided to perform in vitro tube formation assay using human endothelium cells on matricial. So these cells on matricial are capable of tube formations, but after 24 hours more or less, this blood vessel network starts to dissociate, as you can see here on those pictures. And apparently the culture of human endothelial cells with LN18 cells derived from spheres stabilizes this network. And as you can see here, the LN18 cells derived from spheres that are here stained with gel, so they are in red. They sit on those vessels and support the whole network. So this is a very preliminary result, and we need to work on that aspect a little bit more. So to conclude, to sum up, our analysis of the DNA methylome and transcriptome of low and high-grade gliomas reveals differentially methylated and expressed genes, including new gene DMRTA2. And this gene is methylated and highly expressed in GBMs, and its expression correlates inversely with patient survival. This gene and protein DMRTA2 is overexpressed also in glioma spheres enriched in glioma stem cell, and its knockdown reduces the sphere formation capacities. DMRTA2 localizes around endothelium, and cells derived from glioma spheres increase the formation of new vessels in vitro. And with this, I would like to thank all the people that contributed to this work from our laboratory, all the doctors from different hospitals that collaborate with us from whom we get glioblastoma patient tissues. And thank you for your attention.
Thank you very much, Marta, for this interesting presentation. Are there any questions? Just to check the chat. Okay, so you say that DMRTA2 is present in pericytes and involved in tubes formation, or
I am not sure yet if these are pericytes or these are just glioma cells that acquire some pericyte-like properties. So we don't really have proof that apart from the alpha-SMA and the alpha-SMA expression and the localization around blood vessel, we don't have any proof that these are pericytes.
Do you have any idea on the mechanism on this tubes formation? What is in fact the role of the gene or protein?
I have absolutely no idea yet. We need to. This is very preliminary result, and actually, the DMRTA2 is very unstudied gene. And when we studied this project, we had absolutely nothing to start from in the literature. And from the apparently in the last few years, there are some new publications that support the role of DMRTA2 in neurogenesis. But nothing in cancer, and I have absolutely no idea what kind of function this DMRTA2 can have in the blood formation. So this is a mystery so far.
Okay, we have a question from Tamara. Tamara, please.
Yes, thank you. In your essay on tube formation, have you also used brain-derived endothelial cells or only HUVEC?
No, only HUVEC so far.
Only HUVEC.
Yeah.
And you know, as far as I remember your slide on the in the tissue, you have seen this gene also associated with SMA in the tissue, and this was around the vessels, right? Because it was like cross section of the vessel. And so I believe that, or based on what we have also worked on, this should be stem cell niches around the arterioles, and they should express pericytes and also stem cell markers at the same time if stem cells are associated with this arteriole as in the niches. So that would be my explanation of your results.
Okay, I have a question for you in this case. So what kind of stem cell marker would you recommend for us? Because we tried OLIC, we tried NG2, and as far as I remember, we also tried prominine, and nothing worked. So
no, we tried just a CD133, we tried SOX2, and that worked.
SOX2 is very much expressed in glioblastoma tissue, so there is a lot of glioblastoma tissues that express SOX2.
Yeah, but not specifically around the vessels. That would be stem cells, because we used SOX2, CD133, and all this should localize in effect. So okay. You can express this addition to your marker. Yeah, okay, very interesting. Thank you.
Okay, thank you, Tamara. And there's another question from Sylvia on the chat. Did you try to express DMRTA2 in GBM cells and check if their ability to form spheres is increased or induced?
Not yet. Not yet.
Not yet.
Not yet. Yes. Yes, but this is something to do. Yeah, of course.
Okay. If there are no other questions, thank you once again for your presentation.
Thank you.
And thanks to all presenters. We have now coffee break, and well, the next part of the session starts at 10:50.